Hi, everyone. Thanks for coming. I am Madison Huebner. For those of you who don't know me, there's some basics. I'm the program manager for the IOE, and this is our seminar series. And today we have Dr. Brent Peyton, who I assume most of you know because I don't recognize you, so I think you're here on his behalf of his institute. So Brent is a professor in the Department of Chemical and Biological Engineering here at Montana State University. And he's also the director of the Thermal Biology Institute. And I was going to do a plug for your seminar series as well, but maybe you guys.
I've got a slide for it.
Do you? Okay, I'll let you do it
I
then.
have a plug for our seminar series.
Okay, perfect. So Brent has over 35 years of experience studying biological systems. He's focused on characterizing microbial processes in natural and engineered systems, including extremophiles, heavy metal biotransformation and toxicity, and growth of algae and fungi for biofuel production. And Brent actually did his PhD here at Montana State University, and then he worked in the bioprocessing research group at Pacific Northwest National Laboratory for 5 years. Following that position, he was tenured at Washington State University, and he returned to MSU in 2005, and since has authored and co-authored over 145 peer-reviewed publications and holds 5 patents in environmental biotechnology, which I thought was cool. And Brent has also been leading the Extreme Biofilms NSF NRT here at MSU, which has been extremely successful. And additionally, he's been a really invaluable member of the IOE community, especially over the past few years, and I've really enjoyed getting to know you and work with you.
Thank you.
So yeah, take it away.
All right. Well, thank you guys for coming and giving up your lunch hour on such a lovely spring day. I'm going to give kind of a lighthearted talk, go into not a lot of depth, but a lot of different kind of results just to stimulate some ideas. I'm happy to talk in depth on anything later if you want to do that. So here we go. So Yellowstone's the result of a huge volcano. We're basically living very close to a volcano. It's a result of the North American plate moving over the top of a hot spot. And so the hot spot's basically staying in the same location, and the continent is moving over the top. You can see this if you look at a number of eruptions that have happened in Yellowstone. So back 16 million years ago, 15, and so some of the more recent ones up here by Yellowstone, 630,000 years ago. And the eruptions are roughly every 640,000, so maybe in the next 10,000 years sometime. But it's not an exact science by any means. But you can see that it's been doing this for a long time, and eventually Billings will be down here over the hot spot, I think, and so, but we'll all be long gone by then. Yellowstone has a magma chamber under it that's about the size of 11 Grand Canyons, full of magma that contributes to these giant eruptions. The eruptions, some of you, a lot of times I give this to students and they're not around it when Mount St. Helens went off. But Mount St. Helens in 1980 was a 0.24 cubic mile of material came out. The biggest Yellowstone eruption was 600 cubic miles of material that came out of the volcano. That was the 2 million years ago one. And so to put that in perspective, I can't even hardly picture 600 cubic miles of material. If you drive from here to Seattle, 50 miles on the interstate, 50 miles on each side of you, 50 miles on each side of you would be 46 feet deep with lava, okay? It's a lot of material. 21 feet deep on all of the entire state of Montana. So hopefully you have a 3-story house. What's left now is the Yellowstone caldera. It's around 40 by 30 miles long. I'll just X that. Hopefully we're sharing some audio. There's 11,000 thermal features, so about 55% of the world's hot springs, and less than 2% have ever been characterized for microbial life. So there's 98% of the hot springs in Yellowstone that still need somebody to go sample them and figure out what organisms are living there and what they're doing. The Thermal Biology Institute was established in 1999 to start this kind of studies. So we've been around 35 years, 25 years, 35 years. No, 25 years. And I'm thinking of the CBE, which is 10 years older than the TBI. And so essentially things have changed a lot in Yellowstone. Back in the day, it was feeding little bears peanut butter and jelly sandwiches out of the car windows. And now, they're more natural and being tracked with satellites and GPSs and things like that. Same with the hot springs. Used to be kind of gee whiz, there goes Old Faithful again. And now people look at the hot springs a lot differently than they did at the time because they didn't think there was any life in the hot springs. But now each of these springs is a different ecosystem with different organisms and different conditions, pH, temperature, ionic strength. You see organisms, photosynthetic organisms on the edges. These colors are different. There's probably 1,000 different organisms in these two springs, even though they may be inches apart. And we see things like archaea and bacteria, fungi, photosynthetic organisms, and all their viruses are all living in these. And again, very few have ever been characterized. We're going to talk about thermophiles, but I just wanted to point out there's a lot of other extreme organisms out in the world. So if this is kind of what we think of the normal Madison River, duck pond kind of lake, thing, then there's organisms that live at very high pHs, very low pHs for acidophiles, cold-loving organisms, organisms that require salt, organisms that require high pressure, live at the bottom of the ocean. So we're just going to talk about one class of extremophiles, but there's a whole lot of organisms out there that have never been characterized and live at very extreme conditions. The thermophiles, they live at extremely high temperatures. So you didn't think you'd be getting any homework, but if you go home tonight, turn on your hot water tap full blast, that's coming out at about 60 degrees C, okay? Most people's hot water heater thermostats are set at about 60 C. That's kind of in the cold range for a lot of thermophiles. Some of them wouldn't even be able to grow at that temperature because it would be too cold for them. Okay, so some of the extreme thermophiles can only grow down to about 80 degrees C. Some will grow even over the boiling point of water if you keep it pressurized. They also can tolerate other extreme conditions like toxic metals, acids, alkaline conditions. And so again, they can be multi-extremophiles and not only live at high temperatures. This is a little controversial, but a lot of thermophiles show up in a lot of branches of the tree of life. And so a lot of people argue that thermophiles are probably some of the oldest, original life on Earth. But everybody has their own pet theories on that. So there's, you get people that'll argue for iron oxidizer or various organisms. And then just thought I'd throw this theory last time. So why study thermophilic systems? They're basically very unique biogeochemical interactions. Some reactions happen at high temperatures that aren't thermodynamically favorable at low temperatures. And so you got to think kind of different sets of reactions. Better understand the biodiversity of life on Earth, provides clues to conditions on early Earth and evolution. The signatures help us look for life on other planets, give us ideas of what might be possible in other planets. Only 1% of these organisms have ever been grown in a lab. So if you're looking for something with a wide open field of research, 99% out there has never been grown in a laboratory. It's not easy though either. So, and then I think a tremendous biotech potential for society. For things like catalysts and things like that. Half the pharmaceuticals in the US are derived from or patterned after natural compounds. And so As global biodiversity declines, places like Yellowstone and other preserved areas become increasingly important as sources for biodiversity for things that we may not have even imagined we want to look for yet, right? So we may discover in 20 years that there's some new enzymatic catalyst, and if it's gone, obviously we've missed our chance. Okay. So you guys have probably heard about PCR or DNA technology. If you if you watch CSI and they find a hair and they can figure out who did the crime, that's possible because of an enzyme from an organism called Thermus aquaticus, and it's heat-stable enzymes. It allows PCR to happen, polarized chain reaction. It's basically a DNA photocopier, so you can amplify it out. So it allows criminal identification, medical diagnoses. It was used in the Human Genome Project, and it's actually even used to manage Yellowstone grizzly bears. If a bear commits a bear crime, they find hair and saliva, do a DNA profile, and figure out which bear did the crime. Okay. So I'm just going to kind of go through a set of snippets of various projects and things that I think are interesting with Yellowstone. This is a grass called panic grass. Lives right up to the edge of hot springs. And it requires a fungus on its roots to confer that heat tolerance. So it'll grow at very high temperatures, but only if it has a fungus growing on its roots. And it turns out, so this is soil temperature 122 degrees Fahrenheit or 50 C. The panic grass is super happy, just loves it there. You can take that fungus and put it on bean sprouts. Obviously, these beans didn't get the fungus, and these beans did, and so you can transfer that heat tolerance to other plants. And there's a company doing that to try to build up resistance to hot heat waves and stuff that are temporary that might kill crops. And interestingly, it turns out that that fungus converts the heat tolerance, but people discovered afterwards that that fungus has to be infected by a virus to make it transfer the heat tolerance. And so it's a chain of organisms that have to be present to make this work. Again, just kind of showing how complex life can be. I'm going to jump to one of my favorite areas in Yellowstone, which is the Heart Lake Geyser Basin. And here we're standing about 5.5 miles from the road, looking down towards Heart Lake, which Yellowstone Lake is right over there. And it's about 2 miles of some of the most alkaline hot springs in Yellowstone. And so years ago, when I was here on sabbatical from WSU, chose to start sampling here because everybody else was looking at acidic hot springs and nobody was looking at alkaline hot springs, and I had done some other alkaline work. So we have no shortage of volunteers to go on these various trips. We typically do a week-long trip. Been doing a week-long trip to Heart Lake almost every year since 2005. Usually have horses carry our gear in. I like this photo because it's professor, postdoc, that's Dana Skarupa when she first started working with me, grad student, an undergrad, and a high school student, all doing a science project. It turned out that was the high school student did her science fair project on this particular spring. And we often see bears, but we make a lot of noise. People always ask if we're worried about bears. And these are pictures that I took of bears going the opposite direction because we're making noise and singing Disney songs and things like that. And then we've never seen a wolf back there, but we've seen their tracks. Okay, so I started working in that area, but looking at biocatalyzed alkaline hydrolysis. And so this is at high temperatures and high pHs, a lot of organics will break down naturally. And so what we're looking for is, can the microorganisms in these hot springs accelerate those degradation reactions? And so we got a little bit of money from the DoD to look at explosives, and it turns out Catherine Van England did her PhD on that, And it turns out that it does accelerate the degradation of TNT, having microorganisms in there, but not enough to make it worth using that as a bioprocess to destroy TNT. And so we started looking at lignin and cellulose, and actually that's still an area that's still going on. We've never looked at pesticides or looking at using these organisms in alkaline wastewater, but those are open areas of research that people could take up. We started looking at the ability to break down lignin. This is kind of a model of lignin. Lignin's a very complex molecule. And so we're looking for ways to determine whether organisms could break down lignin. You go to hot springs and you look in the bottom, and it's not filled to the top with leaves and branches. It's obviously something in there is breaking it down. And so we found a compound that's this RBBR, Remazole Brilliant Blue, and it's similar to lignin. It's got a lot of aromatics and branches, but it's blue when it's whole like this, but when it's broken down, it goes clear. And so we used it as a screening tool, and every single spring that we tested compared to the blank showed the decolorization. And so this was our first indication that there are probably quite a few different organisms in these hot springs that could break down lignin. And so we tried corn stover and corn cobs and lodgepole pines, and these two we got pretty good growth on by just putting them in spring water and letting the organisms munch on those materials. We started sampling some other springs. This is the Five Sisters near Octopus Spring and the famous Mushrooms Spring where Taq polymerase was isolated. It's just over, I don't know, not too far from there. So it's always an adventure going and sampling these. Again, lots of fun. We focused on two springs, one that we called Tree Fall for obvious reasons, and then a spring right next to it, Five Sisters Spring number 5, that actually that's number 2, that didn't have as much wood in it, and started looking to isolate lignocellulose degrading organisms. And we came up with a organism, Geobacillus thermoleovorans, that grew very well on lignin and cellulose. Consumed oxygen when we gave it lignin, cellulose, or xylose, so the oxygen concentrations went down, CO2 went up, cell numbers went up. And so this is a image, SEM image of lignin with no organisms, no cells on it. And when we inoculate this lignin, you can see that this Geobacillus just loves this lignin. It just gets into every nook and cranny and is breaking it down. So this is my plug for a TBI seminar. Katie Ford out of Bob Kelly's lab is coming up to Bozeman on Monday to look at, give us a presentation on temperature effects and fermentation. They focus on lignin and cellulose degradation, and I'm sure she'll give a good talk on Monday. And then we're going sampling in Yellowstone for a couple of days next week. Okay, some more snippets. This is a study that we did on 3 springs within about the size of this room, maybe a little smaller, so very similar environmental conditions, but and the same pH, but different temperatures. So 44, 63, and 75. And you can see they're very different colors. It turns out that at 72 degrees C, you can't get any photosynthesis above 72 degrees C. And so it changes the community significantly. And I'm not going to go through the Latin names, but my point on this slide is that as the temperature goes up, each of these different colored bars represents a genus of, in this case, Archaea. And you can see that these communities are relatively complex. There's a number of different organisms, maybe 1,000 different organisms, because we've cut a lot of them off. And they change with the years. Sometimes less complex, sometimes more complex. Some of them stay very similar, but again, a lot of bars in these. But once you get above the 72-degree range with no photosynthesis, the carbon source is limited, the energy available is limited, and the community numbers go way down, even though they do change year to year. Here's an interesting spring we call Gabby Spring. Gabby was the high school student that studied this spring first. It comes out at 91 degrees C and flows down here. And it's a pretty slow flow. And as it cools off, it goes from 80 to 71 and 69 there, 76. And so what you see is that white to green transition is almost always right about 72 degrees C. And so you can just walk up to a spring and look at it if you know that and guess very accurately the temperature at various places. Okay, still all of it hot enough to burn you. And then the colder temperature here is typically in the 50s and 60s, the orange colors. So you can just look at a spring now and say, I know the temperature of that spring if you just point to the right spot, that white to green interface. When we sample the DNA of archaea, we can get a few that we can name, but about 50%, 30%, the size of these black dots represent the numbers of organisms or the percent of organisms that we can't really ID even at the genus level. So it'd be like, for example, having a puppy in that corner and an orca whale in that corner and not really knowing the difference, right? So not knowing what to feed it, whether it lives in the water, anything much about it. And so that's kind of how different not knowing, having the genus not known kind of puts that in perspective. And very similar for the bacteria. More bacteria that we can assign names to, but still a high percentage of unknown organisms. Again, lots of work out there to still do. Just to give you an idea of the diversity of environments, even in one location, this is we call the dome. There's 92-degree water coming out up here, and it cools down as it goes down into Witch Creek, which is pH 10 and 55 degrees C. Here's a very unoriginal name, but Spring 2, that is 91, 92 degrees C and intermittently flows and goes underground. This spring we've never sampled because it just looks too dangerous to try to get a sampling pole in there. And people ask if we've thought about drones, but it's just not worth it. There's 10,000 other springs we could look at that haven't been looked at. So again, oh, and then that little guy up there is also very hot, 91. And to put it in perspective as far as the size, all of those guys are about 6 feet tall. So it's a big formation. It's very, I don't know, it's a fun place to work. So we looked at Spring 2, and for years we collected a bottle of water. We filtered some water. We couldn't get any DNA out of it. Finally, Rebecca Mueller and I sat there and filtered 40 liters of groundwater by hand with a hand pump, and finally got DNA out of that spring. And let me go back. I didn't put that skull there. That's not a photoshopped in or anything. It was just sitting there. I thought it made a good picture. When we did finally get the DNA, we found 4 bacteria that we could identify to the genus level and 8 archaea that we could identify, half of them to the genus level. So again, very unknown community. We get an idea of what's there, but a lot of stuff that we can identify down to the phylum. So it turns out this is not all that uncommon. There's been a lot of work. This slide's now 8 years old, almost 9. And this is showing all the bacteria that people know about, mostly through DNA sequencing. And each of these red dots is a major lineage lacking an isolated representative. And so that means that it's a main group of organisms that nobody has ever managed to isolate an organism to study carefully. Same down here. This is the archaea and the eukaryotes that branch off of there. And so again, a lot of organisms there that nobody's been able to grow in a laboratory to study. If I'm not going to spend any time on this because I was running out, I had too many slides, but this is another study that we did, basically looking at trying to grow some of those organisms that nobody had ever grown before by trying different methods. And essentially, we put electrodes in the hot springs with a voltage applied as electron donor and electron acceptor to grow organisms. We left those in place for 6 weeks one summer, and then came back and did DNA extractions on those, and found some organisms that nobody had ever managed to culture before, but we didn't try growing those in the lab. We just took that information, wrote a paper, and called it good. But it's an interesting area of research. Maybe once we're able to make more electricity through solar energy or maybe fusion, there may be options for growing organisms directly using CO2 directly on electrodes for various applications. Okay, I'm going to talk just briefly about some biofuel stuff that we did in Yellowstone. So we also looked at some of these alkaline warmer springs for production of biofuels. So we're sampling in Yellowstone here, and then bring those cultures back and grow them in the lab to try to make biofuels. Under the microscope, this is what they look like. Hard to tell if that one makes biofuels or not, but if you stain it with Nile red, if you stain it with Nile red and look at it under fluorescence, if it's got bright yellow dots like this, then that's lipids, that's oil that this algae has made. And you can even see droplets of algal oil inside of this culture. It turns out when we grow this organism, so this is the Nile red fluorescence, which essentially is a measure of the oil content. Under normal conditions, we grow it. It'd take about 2 weeks to grow up to its full oil potential. But we figured out if you add baking soda, of all things, at just the right time, that time there, you can cut that time in half. And so when we're talking about making algal biofuels and requiring 20 square miles of algae ponds, by adding baking soda, you could get it down to 10 square miles and produce the same amount of biofuel. Arm & Hammer paid for the patent on this, one of the 5 patents. We never made a lot of money on it. We got a bunch of research money, but since nobody's driving around with algal biodiesel in their cars, we never made any royalties on the patent. We also found a diatom that produces very high concentrations. And let me just go back. Diatoms are the prettiest little creatures ever. Basically, they're little like algae that live in silica houses, little glass houses, and just any chance you can to take SEMs of diatoms, I'd highly recommend it because they're all lovely. This one, when we did the Nile red fluorescence, you can see it's just full of each little diatom is full of oil. And sometimes this particular organism made so much lipids that it would blow its shells apart. We'd see them cracked in the oil leaking out. Same sort of thing, the sodium bicarbonate doubled the rate of lipid production and doubled the extent. And when you microwave these cultures, you can see the oil comes out. It's basically olive oil, but actually has one of the highest smoke points, if you're a chef, one of the highest smoke points of any oil out there. But it's not cheap. It's like $18 a bottle. But some chefs swear by it. but nobody's got it in their cars. We did a bunch of scale up studies and finally determined we could probably make biodiesel from algae for about $15 a gallon. It's the best that we could come up with the bicarb added and optimum growing conditions. And at that point, just I decided to get out of the algae research business because it just seemed like it was never going to quite beat out hydrogen and electric vehicles. But it was fun research, again, Yellowstone based. So I want to start to wrap up. I've got a student, John Shikani now, PhD student. He's actually over in Perth, Australia for a 6-month internship with Jeff Pouzon, who's one of the world's experts on free-living amoeba. And it turns out that some of the most pathogenic ones live in hot springs in Yellowstone and other warm climates. In Australia, they have them in their drinking water systems because their drinking water is piped for hundreds of miles in black pipes on the surface of the ground. And so the water gets in the 45 degrees C. Thankfully, we don't have that problem. So Naegleria, there's a lot of different species of Naegleria. So this is work that we're doing right now. And what I'm going to show you is stuff that we're hoping to publish here in the next 3 or 4 months. Naegleria, there's about 47 characterized species. They have different life stages. Some of them are known to be animal pathogens, the Australiensis and Italica. The Naegleria fowleri, you've probably heard of it as a brain-eating amoeba for human brains, and causes the disease primary amoebic meningoencephalitis. That's a tough word to say. But if you do end up getting it, it's a 97% fatality rate and a very rapid disease progression. However, the good news, we only know of 430 cases worldwide. There's probably more because it may not be reported, but it's about 4 in the US, about 4 cases per year. It turns out this little guy lives in warm water, has a variety of different, it can form cysts that are very stable and live over the winter. It can swim around, but if you jump into the water and get water forced up into your sinuses, it can then cross through your sinuses into your brain and become deadly. About 60% of the cases worldwide are from swimming and diving. Most common in the summer when people are swimming and the water's warmer. And then the median age is 14, and most of this is, rambunctious children jumping in and out of the water and getting water forced up into their sinuses. It turns out there's more and more with DNA technology, there's more and more detection. So Nevada, Minnesota, this is a study in Louisiana. All the different colored parishes that you see there had Naegleria fowleri in their drinking water. So it's past the chlorination system, so potentially in the showers and whatnot. And then we plotted, this is built on a different study, but we've added to it. Essentially, starting cases in the 70s and 80s, 90s up to the present. A lot of these blue dots are in the south and not very far north. The red dots are more recent, so it's starting to expand northward and westward. And then this is the latest data we have in the purple. And again, a couple of deaths like 2 years ago at Lake Mead and some in the Midwest. Okay. So it does seem to be moving northward with climate change or global warming. The area that John's studying is this, basically recreational waters at national parks. And so all the way down to Lake Mead, most of what I'll show you here is from Yellowstone and Grand Teton. This is funny, this was colored in my Anyway, doesn't matter. So these are detections by qPCR of Fowleri that we got in the Firehole swimming area, the Boiling River, which you can't really get to swim in anymore because of after the Yellowstone River flood, unfortunately. Lewis Lake, if you didn't know, Lewis Lake has a couple little hot pots on the other side that you can canoe over to on the near the outlet of the river. And then down in the Beckler area, Denanda Falls and Mr. Bubbles, which Maddie has been so helpful helping us sample on a wonderful backpacking trip every year down there. So what we're seeing is we're finding a lot of Fowleri in these recreational swimming areas by DNA. Hold on, somebody's calling me. Okay. Oh, that's why it's colored. I don't usually do animations. Because they're slow and painful. Okay, so this is Grand Teton and also Huckleberry Spring and Polecat, which are recreational swimming areas, although they have signs saying don't swim there. People do. But we use Polecat as kind of a positive control because we've almost never not found Fowleri in that particular spring. And just as a heads up, if you go there to sample, there's a guy named Naked Bob that's always there that'll be there to tell you about Fowleri. And he apparently knows quite a bit about Fowleri. To kind of put it in perspective, this is typical swimming temperatures, 68 to 101. So I keep my hot tub at 101. Chico Sunday was at 101. So these red are the temperatures that we measured when we got a positive detection for Fowleri DNA. And so again, very comfortable swimming temperatures. 68 is a little cold, but we're seeing Fowleri in the temperatures that humans like to swim in. Okay. Do we have any undergrads here? No. Okay, well, I'm gonna make a plug for a class that we teach. So you can tell your undergrads if you do run into any. Dana and I, mostly Dana, does a honors class, Extreme Microbiology of Yellowstone. And in that class, we spend a week doing training, safety training and whatnot. And then the second week of the class, we spend it in the Heart Lake Geyser Basin. But have horses carry our gear in, show the students how to sample hot springs safely, do fireside mentoring. It's really a fun class. So I always like to put a plug in. If you have good students, recommend that they take that course. They get to do an authentic image of science. So after they get the samples back, they get the after they get the samples back, then they get to plan experiments, they get laboratory skills, they spend the rest of the semester a day a week in the lab and a day a week in the lecture. Get to do data interpretation and communicate their findings. And so it's a fun class. We've been focusing on looking for plastic degrading organisms. So discover plastic degrading organisms in Yellowstone hot springs. And we've gotten some nice isolates that we're currently working with from this class of typically sophomores and freshmen. Okay, and so that's that class. And then I also want to put a plug in for our biofilms in extreme environments, NSF research experience for undergrads. So that is, applications are due Friday, March 28th. So this Friday. And we have 10 students that we accept from all over the country, but MSU as well, and a $7,000 stipend, travel allowance, a meal allowance, and they get to work in labs and do Yellowstone and extreme other organism research. Again, Maddie was a host for 2 students that we did a Mars simulation and of biofilms growing on Mars, which was kind of fun. We published that. I've got a lot of acknowledgements. I'm not going to read them all, but TBI has been a great place to work. We've had funding from foundations and DOE, DOD, energy companies. And then this last work with the FALRI is a huge group of people, USGS, actually a very small number of people, but a large number of institutions, USGS, NIH, some of the funding from the IOE. Anyway, a very nice group of people to work with, CSIRO in Australia. And with that, thank you. And if there's any questions, happy to answer.